Okay, welcome to the lecture on Chapter 21, all about infectious diseases of the respiratory system. So first we'll look at the anatomy of the respiratory tract. So it's comprised of the upper respiratory tract, which is all the cavities of the face, the nose, the sinuses, the mouth, and then the upper part of the throat. And we have the lower respiratory tract, which is the trachea and the lungs. And the entire tract is lined in a mucous membrane because it's a cavity open to the outside. And then additionally in the trachea we have cilia, which are extending from the cells lining the mucous membrane in that area. And these cilia will work to trap any inhaled microorganisms or inhaled foreign matter, as they have trapped the bacterium shown in the right part of this figure. And they will then move the trapped matter up towards the throat, where it will be coughed out or coughed and swallowed. So this is called the ciliary escalator. We also have mucus, which is secreted by the cells lining the respiratory tract. So this will trap inhaled matter as well, or inhaled organisms. We have secretory immunoglobulin A, so this is an antibody that is released from the mucous membranes, and so it will bind to antigen and target them for destruction. We have alveolar macrophages, which are phagocytic cells that can attack and destroy foreign cells that will be inhaled. And then we also have the presence of cytokines, which can communicate messages between immune cells and recruit immune cells to the area if needed, and complement proteins. In the respiratory tract, we have quite a lot of normal biota, so there's a lot of microbes that grow in the respiratory tract of a healthy person, and these will all prevent the growth of pathogenic microbes by microbial antagonism. However, in the very lower part of the respiratory tract, in the lung alveoli, we should not have any microorganisms growing. So this is an area of the body that is free from microbes. Okay, the first microorganism that causes disease in the respiratory tract that we'll discuss is Streptococcus pyogenes. So this is a round-shaped bacteria. Strepto tells us that it's arranged in long chains. It's also known as group A strep. It's gram-positive, and in 15% of people, it's part of our normal biota. It causes pharyngitis, which is commonly referred to as strep throat, and in some cases, types of strep pyogenes can cause a more advanced pharyngitis, which is scarlet fever. It also causes rheumatic fever and glomerulonephritis. We're not going to talk about rheumatic fever or glomerulonephritis, so we'll focus on strep throat and scarlet fever. So the way that strep pyogenes is transmitted from host to host is by respiratory droplets. So when a person sneezes, the bacteria can be present in the sneeze droplets and then can be inhaled by a second host. Also, if you come in direct contact with mucus containing strep pyogenes, you can contract the microbe this way. Strep pyo has several virulence factors. So first, on its surface, it has antigens that mimic host proteins, which can allow it to hide from the immune system when it initially enters a host. It also has something on its surface called M protein, which helps the bacteria to resist phagocytosis, and it promotes adherence to host tissue, so enables it to infect the host. And it is coated in a capsule, which would also help it to resist phagocytosis. Streptococcus pyogenes produces a few toxins. Streptolysins can lyse red blood cells, and an erythrogenic toxin, which is produced by the bacteria in cases of scarlet fever, causes a systemic rash and a systemic fever, so it makes the disease much more severe. Typically, the tissues infected are the upper and lower respiratory tracts, and signs and symptoms of pharyngitis would be an extremely sore throat because it's so inflamed, fever, headache, and nausea. Scarlet fever, you have the same signs and symptoms as pharyngitis, but additionally there's a sandpaper-like rash over the entire body, a very high fever, and this disease is much more severe and can be fatal. To diagnose pharyngitis or scarlet fever, you would take a pharyngeal swab. So the physician would take a Q-tip and would swab the back of your throat. And then we do a rapid diagnostic test. So in a matter of minutes, you can tell if there are surface sugars that are present on Strep pyogenes bacteria, and there would be antibodies in the test that would be designed to react against the Strep pyogenes sugars. So if those antibodies react, then we can determine that the patient has the microbe in their pharyngeal swab. And then confirmation can be culturing on blood agar to see if there is lysis of the blood agar in the pattern you would expect. But this takes 1 day, so often the rapid diagnostic test will be enough for the person to be diagnosed with strep pyo infection. And prevention is hand washing and penicillin, which is an antimicrobial drug for treating the bacteria. So when you have pharyngitis, your throat is very sore and swollen, as shown on the left. On the right, we have a picture of the bacteria. Especially important to note are the M protein, which prevents phagocytosis, and the capsule, which does the same thing. When a person has strep pyogenes that causes scarlet fever, this is a strain of the bacteria that has gained a gene for making the erythrogenic toxin that makes scarlet fever so much more severe. And the way that the erythrogenic toxin was acquired by the bacteria is through transduction. So a bacteriophage virus has infected the bacteria, inadvertently injected a bacterial gene for erythrogenic toxin. So now the Streptococcus pyogenes on the right has its genome in pink, and additionally has the erythrogenic toxin gene in blue. So this microbe is much more virulent and causes a much more severe disease. And this picture of a patient on the right is a person with scarlet fever, and this person has a very severe fever and red rash, which is making their face discolored. Okay, the second microbe of the respiratory tract that we're going to discuss is the influenza virus. So important points on this is that it's an RNA virus, seasonal circulation, so it spikes every winter with varying virulence. So some years flu is much more severe than others. 5 to 20% of the world population get the flu each year, and it's fatal 1% of the time. So the transmission mode is respiratory droplets. Also fomites can transmit flu. And virulence factors are its ability to mutate very rapidly, especially its hemagglutinin and its neuraminidase spikes on its surface, which we're going to talk about in more detail in the next slide. So infected tissues are the respiratory mucosa or respiratory mucous membranes. The virus is very good at binding to those cells coated in cilia along the respiratory tract and sticking there. There is a 1 to 4 day incubation period, so you're exposed to the flu and you start feeling crummy a few days later. Signs and symptoms are headaches, chills, dry cough, body ache, fever, stuffy nose, sore throat, and fatigue. And in some cases, it may lead to viral or bacterial pneumonia because the immune system responds to the flu virus, and so other microbes have a chance to take over and make the patient ill. Diagnosis: there are several rapid influenza tests that can be completed in less than 24 hours. So PCR tests for the presence of the flu RNA. ELISA would test for the presence of flu antigens, and immunofluorescence would be a way of staining flu particles so that you can visualize them really easily. And then you can also culture flu, but this takes 3 to 10 days, so usually this is not used. Prevention and treatment is the flu vaccine that is made from a combination of the most prevalent flu strains from the year prior. And then if you catch the flu early, you can take Tamiflu, which is an antiviral. Okay, so here's a picture of the flu virus. I want you to especially note the RNA genome in the interior. On the exterior, we have the two main virus antigens, which are hemagglutinin and neuraminidase. So hemagglutinin is important for attachment to host cells, and there are 16 subtypes. Neuraminidase is important for release and budding from host cells, so there are 9 subtypes. And the flu strains are named by their hemagglutinin and neuraminidase subtypes. So one of the most famous flu viruses in recent years was the H1N1 flu because it had type 1 hemagglutinin and type 1 neuraminidase. So one of the virulence factors of flu is that it is constantly changing. It can undergo major genetic changes, which are called antigenic shifts. So these are changes in the hemagglutinin or neuraminidase spikes that make them significantly different. And antigenic shifts are due to genetic recombination between two different flu strains infecting the same host cell. And once they're in that host cell, they recombine and form a new version of the flu. Antigenic drifts are smaller changes in the HA or NA spikes. So these are probably caused by point mutations in the RNA genome, so just a single letter change there that would confer a very slight change in either of these spikes, which may be only one amino acid difference. And this will allow the virus to avoid IgA antibodies because it can slightly alter, so the IgA antibody can't bind as well to this slightly altered form of the flu. So shift is big genetic changes and drift is small genetic changes, but both are constantly happening. So this figure explains an antigenic shift event. So what we have is a flu virus that is found in ducks as a reservoir, and then a flu virus that's found in humans as a reservoir. If both of these viruses are able to infect a pig, because this is a zoonosis, this flu can go from animal to human. In the pig, the duck and the human flu virus will recombine to form an entirely different virus in an antigenic shift event. Okay, the last microbe we'll talk about in chapter 21 is Mycobacterium tuberculosis. So this is a gram variable bacteria. The reason it's gram variable is because if you do a gram stain on it, it will sometimes look positive and sometimes look negative. For that reason, it has to be stained with a different technique called acid-fast staining. So Mycobacterium tuberculosis bacteria is positive for acid-fast staining. It grows very slowly, and part of the reason why it is gram variable and is that it has lipids and wax in its cell wall. So this affects the ability of gram stain reagents to penetrate the cell. So it causes tuberculosis, which we'll abbreviate in some spots as TB. The mode of transmission is respiratory droplets. Its virulence factors are its waxy surface because this enables it to survive inside macrophages by blocking phagosome formation. So although the macrophage attempts to ingest the bacteria prevents phagosome formation inside the macrophage and just is able to exist in the macrophage cytoplasm. This bacteria also elicits a very strong immune response, and it's the immune response that causes a lot of the disease symptoms. The tissues infected are typically the lungs, so the lower respiratory tract, but it is possible that the infection can disseminate or spread to any organ in the body. So it's estimated that only 5 to 10% of infected people develop clinical TB. This bacteria is very good at infecting people, but in a healthy person, it often won't cause disease. The signs and symptoms are that tubercles form in the lungs, which are pockets of infected macrophages that are enlarged, and these pockets are surrounded by immune cells attempting to respond to the infected macrophages. The interior of the pocket the macrophages can start dying, and this forms a hollow area that then heals in a calcification, so it becomes like a hard, bony part in the lungs, which make lung functioning very difficult. There is violent coughing, greenish or bloody sputum, which is what you cough up if you have a really bad cough, fever, fatigue, and weight loss. And so in primary TB, the disease is limited to the lungs. In secondary TB, we have reactivation of a dormant microbe, so the person had TB, got better, and then they have a reactivation because some of the bacteria weren't cleared. And then in disseminated TB, the disease spreads outside of the lungs to other tissues. So diagnosing TB can be done with culturing the bacteria, which is very slow because they grow slowly. We can do a tuberculin skin test, which takes 48 hours to see the results of. Or you can take the sputum and you can do an acid-fast stain to see if you have any acid-fast M. tuberculosis present, and a chest x-ray to look for tubercles. So prevention and treatment is to limit exposure, so people who have TB are limited and are unable to go out into public spaces because they can cough and spread the disease. There is a vaccine that is not used in the United States, mostly because we like to be able to track exposure to TB, which is hard to do if everyone's vaccinated. If you do have TB, minimum of 2 all the way up to 24 months of drug treatment. And in mildly drug-resistant or MDR TB, you have to do 1.5 to 2 years of drug treatment. In extensively drug-resistant TB, there are very few treatment options, and it's often fatal. So shown here is on the left the culture appearance of Mycobacterium tuberculosis, so it grows in these very bumpy columns. And then we have an acid-fast stain on the right showing the broad bacteria. And then reminding you that this bacteria infects the lower respiratory tract and can get into the lung alveoli. This is an x-ray of a person with TB, so the darkened area in the box is an area of infection, and so this is a tubercle that has been formed in their lung. One of the ways to diagnose TB is the MAN2 skin test. So you inject purified Mycobacterium tuberculosis protein. So you don't inject the bacteria, but you inject antigens or proteins from the bacteria. Then 48 to 72 hours later, the patient returns to see if they have a welt that's raised around the area of injection, and this welt is called an induration. If the induration is greater than 5 millimeters to 15 millimeters, depending on if they've been exposed and whether or not they have a healthy immune system, this would be considered a positive TB test. So this person has been exposed to TB or has TB. The reason that the induration happens is because the patient has memory T cells that are primed to respond. So when you inject the TB antigen, the memory cells respond very strongly and you get the raised welt. If you get a positive TB test, it would be followed up by an X-ray or a CT exam, and then also acid-fast staining of the sputum and culturing of bacteria to confirm. So treatment of tuberculosis is going to be various antibiotics, which can be challenging because a lot of the bacteria live inside macrophages, so they have an intracellular lifestyle. Generally 6 months of total antibiotic treatment, and it's often recommended that this is directly observed therapy or DOT, meaning you go to a healthcare facility to take your antibiotic, because taking them regularly and at the same time every day is very important for ensuring that the TB doesn't persist in your body or become drug resistant. If the TB that you're infected with is antibiotic resistant, mildly drug resistant or extensively drug resistant, it means longer antibiotic or very few effective treatments, unfortunately. The vaccine is a live Mycobacterium bovis, which is a related bacteria, but it's been weakened or attenuated. It's 0 to 80% effective in preventing TB, but it's not used in the United States because we're lucky to have very low prevalence of TB, and so we are able to track when people are exposed and contain them, where if we give everyone the vaccine, it's very difficult to track exposure. That concludes Chapter 21.